website into useful high-value products. This could be food and food supplements, chemicals, pharmaceuticals, bioenergy, and we hope to do that in a cheaper way, much more cost efficient, and also in a more sustainable, environmentally friendly way. And how do we do that at SGI? So we utilize the design build test cycle for synthetic cells, and I will mainly focus on the building part. But the idea is that you design your genome, synthetic genome, you build it through sub-assembly stages. As I said, usually our complete genomes end up in E-cyclomycin base. So far that's been the most efficient host for a full genome assembly. And then you test for viability by transplantation. So I will start now with the in vitro DNA assembly tools that I've been working on and developing. And here is just an overview of the current DNA assembly technologies. It doesn't include every method that's out there, and a lot have been developed and published recently. However, it does show some of the major methods. The in vitro methods can be subdivided into two groups based on the technology. If they are based on the glucose restriction digest ligation principle, and if they're based on the in vitro homologous recombination. And as you can see, there are multiple different methods here. The restriction digest and ligation method has been around for nearly 40 years. And it's a very simple and straightforward method, and I'm sure that many molecular biology labs are still using this method. It's very straightforward when you want to clone a single gene into a vector. You use restriction enzymes for that to open up your vector and ligate your gene of interest there. And it works, as I said, well for one fragment. However, for multiple fragments and with the increasing size of the DNA you want to clone, you getting into problems finding appropriate restriction enzyme sites. And for large DNA assembly, this method obviously does not work. And with the advancements in synthetic biology, metabolic engineering, and genome engineering, obviously we need better methods, more efficient and more accurate, and especially methods that would allow us to assemble very large DNA constructs genome size. So, I'm sorry, I'm back now. The current methods we're using at SGI have been developed by Dan Gibson and his colleagues at JCBI. And most of you probably are familiar with the isothermal one-step Gibson assembly approach. It's a pretty straightforward assembly reaction. It can assemble multiple fragments in a seamless way. It's highly efficient, and it results in a fully repaired, covalently closed circular recombinant molecule, which can help you in further downstream applications. You can amplify your sample fragments, or you can even rolling circle amplify them. This assembly method has been demonstrated to work on a very large scale of DNA constructs. It's been used to assemble in vitro the mycoplasma genitalium genome, which shows what the potential of this method is with large constructs. And another version of this method is when you start from oligonucleotides, so you can start from those to assemble your DNA cassettes, and then they keep on being assembled in a hierarchical way till you get to your final construct. And this has been demonstrated for the chemical synthesis of the mouse mitochondrial genome. So there is another method that maybe you're not as familiar with, and it was just recently commercialized. It's also a method developed by Dan Gibson and JCBI. This is a two-step thermocycled assembly, and actually that was used for the first synthetic genome, the mycoplasma genitalium. It's a very efficient method. It's not as straightforward as the one-step because it requires two-step. However, it's a very robust method with very high efficiency, and especially for people who are new to the field and are trying to learn to use this assembly, Again, it's like the first method, it results in fully repaired covalently post structures, and as I said, it's been recently commercialized by SGI. So, how do we apply these methods at SGI to improve our assembly capabilities so that we can speed up the process and build genomes much faster and much cheaper? So one way to do that is if we actually eliminate the biological host, which allows us to save a lot of time. We're trying to do this using Gibson assembly. So if you have two fragments, you assemble them into a vector, it's just an example. And then your assembled construct, you can use in a PCR amplification to amplify your assembled genes. Of course, there are limitations to using the PCR amplification method. With the size, it gets more challenging and you have more errors. And what we became interested in is using volume circle amplification to see if we can amplify in vitro assembled constructs and see how large we can go and what the fidelity will look like. This idea has been around for some time. Already in 2005 at JCVI, they demonstrated this, and later Dan Gibson, with his method, demonstrated that his assembled construct can be amplified by RCA. And so here is a little bit about what RCA is. So this is a isothermal amplification of circular DNA by the 529 DNA polymerase. It has a very high persistivity and very high fidelity. It has strong strand displacement activity, which results in your product being tandem repeats, multiple tandem repeats of your target. It gives you very high DNA yields, even if you start from low amounts of template, and even if your template is crudely purified. So all of these advantages led us to believe that maybe this is a method we can use. The most used version of Rolling circle amplification actually, and the commercial kits that are out on the market, they're using random hexamers to prime the amplification. So that method is called multiple displacement amplification, and what it allows you to increase significantly your DNA load. And this has been very successfully used for whole genome amplification from single cells. It's a very powerful tool. However, with every method, there are some disadvantages. And the problems that you might encounter, the presence of some template-independent amplification, you might get some background. And this could be due to multiple reasons. You might have DNA contamination, or this could be side products from the random hexamers, or your template concentration is very low. So how did we envision using this in our cell-free cloning platform? So again, the purpose of this exercise was more or less to see if we can significantly speed up the process of putting fragments together, DNA fragments together, by eliminating the host, so you don't depend on the cell growth, the recovery of your host. In this way, you can avoid any toxicity or instability issues. It's a really fast method. It's very amenable to high throughput and automation. And we did test it and found out that there were some challenges, and these were involved with when you go further into increasing your assembly construct size, then the efficiency significantly dropped, and also we found out that the accuracy was not as high as when you propagate your DNA in a biological host. So the idea behind the workflow is you have your multiple fragments, you assemble them by Gibson. You don't really need a vector host platform here. Then you rolling circle amplify, and with a single restriction enzyme digest, you can linearize your assembled fragments. Then you go to the next round where they assemble with other overlapping DNA fragments. You assemble that in a circular construct and rolling circle amplify it, linearize your fragments, and you keep on going. And as I said, at certain size, you come into the challenge that the efficiency is not as high, the fidelity is not as high, and you have to move into a living host. So the second part of my talk will be about using in vivo DNA assembly tools and what are the host platforms that we are currently using for this. So there are three major hosts that we use for cloning large DNA. The E. coli host is the one that is mostly used in traditional cloning procedures. With the lambda recombination system, it allows you to clone your gene, it allows you to engineer it inside E. coli. However, as far as size, this platform doesn't really give you the large size assemblies that this platform will provide you with. Bacillus subtilis is another bacterial host that is used for assembling of large pieces of DNA. And pretty impressive work has been done with this host by Itai and his co-workers. They have managed to clone 3.5 megabases of the Synaptocystis genome into Bacillus. So it shows that this organism has a very large capacity to take exogenous DNA of such size. It has a natural homologous recombination machinery, which is very efficient. And those bacterial host systems give you an advantage that they go fast. You can quickly purify large amounts of DNA of very high quality. With E. coli, the size of your exogenous DNA can go up to 500 kb. With Bacillus, as I said, Itai and his co-workers have proved you can go up to 3.5 megabases. However, in both platforms, this assembly process is happening in sequential iterative stages, one step after the other, to elongate your constructs. And in Bacillus subtilis case, these fragments of about 800 to 900 kb were integrated into the Bacillus subtilis genome. And the problem with the systems is that at one point you might get into a toxicity issue where you can't keep going using this platform. And so now move to the Saccharomyces cerevisiae host, which has a pretty impressive homologous recombination system. And Dan Gibson has already demonstrated that you can assemble in one transformation step a full bacterial genome from 25 overlapping DNA fragments in one step, which is much faster than the iterative sequential assembly process with the bacterial hosts. So it does assemble very efficiently overlapping DNA fragments. And how big can we go? So far we're around the 2 megabase size. Surely we can probably push that limit. And then another advantage is that it has a different expression system, and usually you don't have these toxicity issues using this host. The problems with it, of course, as I said, you always have some pluses and some minuses in every method and tool you use. That it doesn't grow as fast, and you don't get as much plasmid DNA milled out of it, and the quality is not as good as from E. coli, for example. And then if you want to clone large sequence into Saccharomyces cerevisiae that is very GC rich, you might have to introduce yeast origins of replication at every 100 to 150 kb fragments. So I will mainly focus on using the Saccharomyces cerevisiae host because my talk will be focused on whole genome assemblies and engineering. So there's several ways you can assemble genome, bacterial genome in yeast, and I will try to explain those here. So one way is if you insert an yeast factor that carries the yeast origin of replication selection marker into your microbial genome, and this can happen either through random transposition or site-directed homologous recombination. In this way, you will have the yeast factor integrated, and then the genome can be purified and transformed into yeast spheroplasts. The purification methods are not as simple, especially with large bacterial genomes. You have to embed those in agarose plugs. However, a new method was developed by Bob Karas and his co-workers at JCVI, can actually transfer your genome without having to purify it with cell fusion, and that will work very well for large bacterial genomes. Of course, if you don't have the option of transforming your organism and you can't really do that, then you have another way to go around this. You can purify your microbial genome. As I said, you usually do that in an agarose plug so you can protect it from shearing. And then you have to linearize it and co-transform with an yeast vector that has homology to the ends of the linearization site, and then transform that in yeast spheroplasts, and yeast with its homologous recombination machinery will put the two together. And a third way is when you want to build your synthetic genome from multiple fragments. These could be synthetic fragments or fragments from your microbial genome. They can be transformed in one step into yeast. And yeast, as I said, with its pretty impressive capacity of homologous recombination, can put them together with the yeast vector into a fully assembled genome. So we'll start talking a little about this approach, and then I'll move later and talk about some of the other options. So at JCVI, a lot of genomes were synthesized and assembled in yeast and activated, and all of this knowledge has been transferred to SGI, where we are trying to implement those methods and improve them and further develop. So usually our workflow of building genomes starts with oligonucleotides, which get assembled into smaller size DNA cassettes, and then you keep on assembling in vitro using the Gibson assembly methods till you get to a size where you move into your in vivo host Saccharomyces cerevisiae. And this process has worked pretty well. However, there's some lessons that we have learned, some challenges and problems that we have tried to address to some extent at SGI. So when the Mycoplasma microides genome was built, the problems that were encountered were that basically they couldn't really propagate fragments over 10 kb in E. coli. This is a very AT-rich genome, and it turned out that those fragments are toxic to the E. coli cloning host. So they had to move all of this into yeast, assemble them in yeast, but then what Dan and his team found out is that purifying those fragments from yeast is not trivial. You get very low DNA yields, you have very low quality of the plasmid prep that you have to further purify, and if you want to use these fragments further from further assembly steps, this might be a significant problem. So how did we try to address that? So one way, the traditional way is, if you're at the stage where you have an intermediate genome assembly intermediate of that size, about 100 kb, in E. coli is not an option where you can go to and propagate your DNA and get sufficient amount and high quality, very pure prep. Then your options are you either do a large-scale plasma purification from yeast, or you try something else. So the large-scale purification is a very laborious procedure. It's very time-consuming, and you get very low yields, as I mentioned before. And you need to further purify those preps in order to get a good enough transformation in assembly efficiency. So what we try to do is, can we use Raman-Sur quantification in vitro to amplify those intermediates, which would allow us to, in a very fast and efficient way, to generate a lot of DNA, which could be then assembled further in yeast. Of course, the questions we asked ourselves were, how big can we go? How large can we amplify in vitro? And what about errors? And so first is, Well, let's try and see if we can amplify something that's close to 100 kb in size. And this is a back DNA of about 96 kb, purified from E. coli, where we try to amplify with the long circle amplification and just see if that is something doable and easily achievable. And so we were successful in this. We managed to purify a good amount of this construct, which we sequenced and then restriction digested to confirm the size and the integrity of the construct. And then we actually showed that we can use it in downstream applications like cloning and yeast transformation. So the current RCA methods have shown that this is very doable even on such a scale of 100 kb size. However, purifying DNA from E. coli is one thing, and purifying plasmid DNA from yeast is a different problem. So the question was, can we actually do that from yeast? And so here is an example of a 97 kb circular yak DNA, which was purified from yeast. And the challenges we were facing, of course, were, well, we have a DNA prep that is still contaminated with yeast linear chromosomal DNA. Very low amounts of template in our prep, so we'll be able to do this. And the answer is yes, we managed to amplify this construct, but what we notice is that you actually end up with a significant background. This is a non-specific amplification happening, and it wasn't really a surprise with having in mind the quality and the amount of our template. But we did manage to get enough DNA, good enough quality DNA to be able to sequence and then to take those 100 kb intermediates, genome intermediates, and then put them into yeast to assemble the full genome. So we managed to do that using this method. And in summary, what we learned from this is that yes, we can do this, and it's much easier and much faster than the large-scale prep from yeast. It is compatible with the homologous recombination in yeast and with Gibson assembly. You can do a size of 100 kb, but the problems are that the fidelity is not as high. We encountered an error rate of about 1 error of 27 kb amplified DNA. And again, the fidelity is not as high as propagating in a biological host. As far as assembly efficiency, using RCA amplified DNA, we found out we are somewhere in the middle. We have a better efficiency than using plasmid DNA from E. coli, but not as high as when you use PCR amplified fragments for assembly in yeast. The accuracy is again in the middle. It's not as high as using plasmid prep from E. coli, but it's better than PCR amplified fragments. And so now I will move to implementing how we implement a new technology, the CRISPR-Cas9 system. And I will just cover a few basics. Again, I'm sure most of you are familiar with it. Sorry if I'm repeating things you already know. But this is a RNA-guided gene editing platform. It's part of the adaptive immune system, type 2, and it consists of a Cas9 endonuclease purified from Streptococcus pyogenes. This is the most commonly used one currently, which is targeted by the guide RNA to a genomic target sequence of 20 random nucleotides, followed by the protospacer adjacent motif signal consisting of NGG, which you need in order for cleavage to occur. The guide RNA can be introduced as the natural dual guide RNA complex, which is made of the CRISPR RNA and the transactivating CRISPR RNA, or you can use a chimeric single guide RNA. And in my experiments that I'm going to talk about next, I most of the time use the chimeric single guide RNA. So this is just a brief scheme of how you can combine Cas9 with Gibson assembly in your cloning experiments. One advantage to using Cas9 is that it allows you more flexibility compared to restriction endonucleases. You have more of a choice where you want to cut. And the efficiency is pretty high. There is multiplex capacity, and the guide RNA is introduced as an in vitro transcribed RNA. So you can linearize your vector with Cas9, and then after the linearization, you can assemble the insert with Gibson assembly. Or where it's mostly advantageous is if you have a large insert, larger DNA construct that you want to linearize, and you don't have suitable restriction enzyme sites to use. Well, Cas9 could be a way to do this. Might give you more flexibility. So you can choose your target sites, digest with Cas9, and then assemble using Gibson assembly. So now the focus of my work was actually trying to see if the Cas9 system could actually help me with the genome assemblies. And how I can improve that and maybe do things better. So one way where I saw using Cas9 as an advantage was the linearization of my microbial genome. So previously this was done with restriction endonucleases, and this approach using Cas9 is again the idea, well, if I don't have suitable restriction enzyme site, and can I have more flexibility in choosing my target using Cas9? And so this is the first thing that I'm trying to do. I linearize my genome with Cas9 and then co-transform with yeast and see if I can assemble that. And so you can do that in vitro where you purify your genome, as I said, in agarose plugs. And then you digest with Cas9 and the guide RNA inside the plug, and then you co-transform with your vector the yeast spiroblasts. And the homologous recombination in yeast assembles your genome. And that works pretty well. And then another option is you can do that entirely in vivo, where you introduce an expression cassette of Cas9 and your guide RNA together with the purified genome and the vector. The Cas9 in vivo linearizes your genome, and then you yeast assembles it with the vector. So once you have the bacterial genome clone in yeast, well, how else can you use Cas9? Well, something that Cas9 is very much used for is genome engineering. So here is an example of how once I clone the bacterial genome in yeast, how I can engineer and edit that genome using CRISPR-Cas9. So we've managed to show that we can do simultaneous insertions and replacements of donor cassettes that don't have selection markers inside yeast with the CRISPR-Cas9 system. We can do seamless deletions of genes, and this is a huge advantage over the traditional genome engineering methods because you actually don't have to introduce selection markers. You don't have to deal with recycling those or coming up with more selection markers. You don't need to use counter selection markers, and it really speeds up and simplifies the whole process. So this is something that works very efficiently in our hands, and we are continuing to improve and work on it. Now that I showed you how you can assemble and engineer this genome, so what are the applications and what are we doing with it? So our current platform is you assemble your genome, you can use Cas9 for that, and then you can engineer it inside yeast, and then you test for viability for transplantation in the recipient cell, and then you keep on going this circle to get to the synthetic genome with the desired functions. The advantage is you can do this engineering inside yeast, especially if you're working with genetically intractable microorganisms, which cannot allow you to do that. So you can do that all in yeast and use the very efficient and well-established genetic tools in yeast, in the yeast platform. You can have the genome silent there, it's non-functional, and you can study gene functions, you can study genetic interactions and genome architecture using all these genome engineering tools that are available in yeast. You can work with pathogenic organisms and remove virulence factors and even generate attenuated viral vaccines. So there are multiple applications for this. And one example how we do that is by using our platform with the Mycoplasma microvirus genome. We try to identify gene functions, and if we know what gene we're interested in, we use well-characterized genes from other species to replace that gene, and using the Cas9 yeast platform, and then we transplant those into the recipient cell, and if the genes that have replaced the one we're interested in complemented, we can get transplants, otherwise there is no growth. So this is what we are currently using, and it works very well in our hands. Of course, we would like to extend this genome activation platform for more microorganisms and make it a broader application, and that is something we are currently working on. And so here is a brief overview of other activities my group is interested in and working on. So a major focus is advancing genome design, DNA synthesis and assembly tools, and genome activation, but we also support other teams in the company. For example, Magda is from the vaccine team, and we work closely with their team to help them, especially when they need some advice in DNA assembly methods. And then we also work closely with SGI DNA and help them with their product development process. And all of these methods have been actually automated under BioXP. At least we're trying to get them all there, and you probably will be able to see the instrument here. I heard that they will show it. So these are all the activities we are more or less involved in at SGI, and I'll end up